Hello student, welcome to Navshala. We provide quality education free of cost. No paid batches, no membership. Only free education to everyone. Children, in previous lecture we started Heredity and Evolution chapter of class 10. In that chapter, three important concepts we completed. One, heredity, second, central dogma, and third, transcription. And in today's session we are discussing about the translation, translocation and how mutation takes place in DNA. Let us start today's session. Children, in previous lecture we completed concept of transcription. In that we understood that whatever information present in DNA, that information is copied on RNA. Especially we talked about mRNA. So synthesis of mRNA as per the nucleotide sequence of DNA is called transcription. In earlier lecture we discussed this. So what was the end product of transcription? End product of transcription is mRNA there. So this is your mRNA there. Once mRNA ready, this mRNA will come outside in this way. So this mRNA where formed, where it is produced? It is produced in the nucleus. First question. Second, once the mRNA ready, where it comes? It comes in an outside area called as a cytoplasm. So second important thing, once the mRNA ready, it will move outside the nucleus and comes in the area called as cytoplasm. In cytoplasm, one special structure present called as a ribosome. Now this mRNA will come to ribosome. Why sir ribosome? Why? Because ribosome is a protein factory there. Whatever protein needed in body, this all protein ribosome is making. So if you remember the central dogma concept, we want protein and this protein information was there in DNA. So information from DNA, it is given to mRNA. mRNA will come to ribosome. Ribosome will make protein. Are you getting? So children, you see, we are telling mRNA, M for what? M for mango there? No. M for messenger. Because this mRNA brings the message from DNA to ribosome. Therefore it is called messenger RNA. No, it brings the message, not vada pav. So concept understand. When mRNA move outside the nucleus, it brings the message for amino acids. So this message is in the form of coded there. It is a coded message there. So this coded message from DNA comes to ribosome with the help of our mRNA. So what is the role of mRNA? To carry message from DNA to ribosome. Sir, message for what? Message for amino acids. Now what is amino acids? Let's understand that. Now see children here. In starting lecture we discussed that curly hair is one biological character there. Hair what there? Hair is one type of protein there. Hair is one type of protein there. If I want to make hair, means I am making protein. That's simple. Hair I am making means I am actually making protein. These are protein there. But sir, this protein is made up of anything or? Yes. This protein is made up of small units called as the amino acids. So this is my first amino acid there, this is second there, third there, like that many amino acids joining, then I will get the protein. So protein is a chain of amino acid. So about this information mRNA will bring. But sir, why there is need of mRNA? Message why you want? If you want to make protein, message word, why there is a message word is used? See, message means specific information there. No, message means it's not random anything there. Specific information there. For example, see, the first amino acid which there here, glycine there. Second one what there, isoleucine there. Third valine there. Fourth cysteine there. Again here glutamic there. Like that in each protein, amino acids are present in a specific sequence. Like your school roll number. In your school roll numbers are there, no? Everyone is having 1, 2, 3 like that roll numbers there. If I am saying roll number 20, then a particular boy or girl only will stand. Not everyone. So roll number gives information to you. So roll number is your code there of school. Like that, mRNA brings code for these amino acids. So as per the code, first amino acid, second amino acid, third amino acid is decided. Therefore, we are telling mRNA bringing coded message. Coded message for whom? Coded message for these amino acids. And these amino acids, when they will join, then finally we will get the protein. Protein forms, then we will make the hair. If mRNA not bringing message, then amino acid will not come. Amino acid not there, then protein not there. Protein not there, then hair not there. Hair not there, then full pala there. Getting? So important there, mRNA should bring the message. Okay. Sir, that message which mRNA bringing, what message there in that? Like this we find AUG, CCA, UUG, GUC, like that message there. Now these message which mRNA is bringing, this message for amino acids, one amino acid consists 3 nucleotides. If you see here, AUG, this gives information about one amino acid, that is methionine. You don't think about that names, no? Name is just for your information there. Methionine, leucine, isoleucine, valine, amino acids. That's it. So AUG gives information about one amino acid, methionine. Then CCA will give information about second amino acid, that is proline there. UUG will give information about third amino acid, leucine there. So if you see here, the one amino acid is decided by 3 letters. AUG together decides methionine. It's not that A for you, A1, U for second amino acid, G for third amino acid. All 3 letters together. These letters we called as a nucleotide. How many nucleotides together there? 3. So the code for each amino acid, code for this amino acid consists how many nucleotides together? 1, 2, 3. So code for each amino acid consists 3 nucleotides. Therefore, it is called as a triplet codon. Triplet why? Triple, 3, AUG, 3. That much you are understanding only. Getting? So code for what? Code for amino acid. So code for each amino acid consists how many nucleotide? 3 nucleotides, sir. So that only we called as a triplet codon. How to define? Simple. Code for each amino acid consists 3 nucleotide. Code for each amino acid consists 3 nucleotide, together called as triplet codon. If suppose I am telling you, in this mRNA, how many codons are there? Codons how many there? Check. AUG, 1 codon there. CCA, second codon there. UUG, third. GCU, fourth. AGC, fifth. 5 codon, 5 amino acids will form. Clear? So mRNA brings the coded message. mRNA brings coded message for amino acids. And the code for each amino acid consists of a group of 3 nucleotides. Code for each amino acid consists of a group of 3 nucleotides. And this group of 3 nucleotides we called as triplet codon. How many triplet codons? That number of amino acids will be added. For example, in this diagram 1, 2, 3, 4, 5 codon, 5 amino acids will be added in a chain. Clear? Then sir, only 5 there? No. There are about 20 essential amino acids are there. Means in my body how many proteins are there? That all proteins mainly made up of 20 type of amino acids. That 20 amino acids are having their particular code. For example, UUU, phenylalanine, CUU, leucine, like that many are there. We have to learn this? No. Just I am showing you. What you have to learn? How many essential amino acids are there? 20. By using that 20 amino acids, your all proteins are formed. But remember that everyone is having their code. So any amino acids, if you see, everyone, serine amino acid there, then it is having one code like that. So many codes, so many amino acids. Who studied this? One famous scientist there, Dr. Hargobind Khurana. Indian origin scientist there, did research outside the country. Their main role, their main contribution there to find out this, just now I shown to you, these means for particular amino acid which code will come, for next amino acid which code will come. This all study did by Dr. Hargobind Khurana. Name you have to learn. Second, what is their contribution? Contribution what there? Contribution there in discovery of triplet codon for how many amino acids? 20. Just now we discussed. For that they got Nobel Prize in 1968. That year Pitpat worked there. Now children, mRNA when it comes outside, no? This mRNA has shown only 5 amino acid sequence. 5 codon has shown, no? Just now. But only 5 there are what? No. Thousands of codons are there. 1, 2, 3, 4, ... Many are there like this. 1000 there, you go that side to count. I am showing this much. Okay? So each mRNA is made up of thousands of codon. Sir, if thousands of codon there, then amino acids also will be 1000. 1000 amino acids will be joined. Obviously, because one code, one amino acid. If codons are 1000, then amino acids also will be 1000 only, no? Yes or no? So codon, how many? That much number of amino acids will be there. So long amino acids will join to make that protein. Now sir, this mRNA is having codon according to that amino acids will come. But who will bring that amino acid here? That is also important there. For that we are taking help of tRNA. tRNA stands for transfer RNA. So as per mRNA information, when we are making a protein, we need amino acids. Why sir? Just now we discussed, no? Protein what there? Protein is a chain of amino acid. Amino acid coming, then only I can make protein. So these amino acids, to make that protein, these players will bring. Who are these? 1, 2, 3. These all are tRNA there. So tRNA what they are bringing? See on their head? Amino acid there. Wait, no. This amino acid there. Here also amino acid there. Here also amino acid. So tRNA what doing? tRNA transfer. Padapa? No sir. Amino acids. So tRNA transfer amino acid. tRNA transfer amino acid to ribosome to make that protein. Sir, now suppose here on mRNA we are having AUG. Then sir, this we understood that tRNA will bring the amino acid. But AUG, it will code for something amino acid, no? Then here 1, 2, 3 there. Which is the correct one? Or anyone allowed there? No. Anyone it is not allowed. You recall this sickle cell anemia. In sickle cell anemia, 9th standard concept there. If one amino acid wrong happening, no, you will lifetime you will suffer from sickle cell anemia. You remember or no sickle cell word there? Your RBC shape will become sickle, like this shape happening. Why? At 6th position, like this there, 1, 2, 3, 4, 5th, 6th. Suppose here 6th there. At 6th position, glutamic acid, this amino acid needed. By mistake there, instead of glutamic acid, valine comes. See here? One only change happened. Rest all amino acid sequence has it is exactly same there. Natural, normal there. One only mistake happened. What is that one mistake? That instead of 6th position glutamic acid, valine comes. One amino acid change, one lifetime disease you will get in gift. No treatment, no cure. Lifetime I am telling. Sickle cell anemia. Only normal treatment you can give. But from body you permanently can't remove that disease. So danger, lifetime disease I can get due to one mistake. So like that any amino acid I am adding, chalo, you add here one, you add here. Like this doing, then protein will form, but it will be abnormal. If protein abnormal, you will be abnormal. So here correct amino acid adding is very important there. and that correct amino acids to add, we are having one system. Getting? What is that system? Please see here. tRNA when bringing amino acid, no? tRNA also having one sequence. If you see here, UAC, it is called anticodon. It is called anticodon. Where anticodon present? Anticodon is present on tRNA. Okay, sir. tRNA is having anticodon. Then, sir, this what there? But this we called as codon. This is codon there, codon. AUG is a codon. Sir, confusion not happening then? Codon, anticodon. Who is coming first? mRNA is coming first in this picture. Okay. Then mRNA is having codon. Codon. Front who is leaving, that is anticodon. So its front which there, that is your anticodon. Simple there. Anticodon. Right? So codon is present on mRNA. Anticodon is present on tRNA. Fix here. Codon on mRNA. Anticodon on tRNA. Easy. Okay there. Why this anticodon made here? If you see, this anticodon pairing is very important there. If you remember the base pairing rule, last lecture we discussed, A is always paired with U, G is always paired with C. A with U, G with C. This is called complementary base pairing rule. Check here. Upside U there. So A is pairing with U. So here U there, then downside exactly you will find A there. Check. Right? Now upside A there, then A front who is there? U. See here. Here you will find the U there. G pair with C or C pair with G. C there, then downside C. G there. So according to base pair, complementary there. Complementary. Lock and key type. Lock and key there, no? Key is specific for lock there. Or anyone's key, we can open the lock. Not like that. So anticodon, codon, complementary there. If here matching happening, if it match, then only amino acid will be accepted. And understand the concept. And learning afterwards, learning after understanding there, no? It is pit-pat we are doing. Concept understand. Sir, we are teaching, no? You understand the concept. Don't be, why running behind that learning out of out? Slowly concept understand. Everything, marks automatic will come. You just first listen carefully concept. Concept understood, then learning feeling coming that. Nice feeling that. Concept knowing, learning also perfect. So tRNA will bring amino acid. Correct amino acid K1, know how to check? Matching to AUG codon, UAC anticodon. Match, perfect. Add amino acid. Getting? So tRNA is having anticodon. tRNA is having this anticodon complementary to codon of mRNA. I will repeat. tRNA is having anticodon. Complementary means opposite to what? Codon of mRNA. So like this, can you tell me in this picture, suppose I am asking to you CGA. I give here number, codon number 1, 2, 3, 4, 5. You have to tell that CGA, this is one anticodon there. This tRNA will match with which codon of mRNA? Check. Complementary rule. Remember just now I gave. What is the rule? Complementary rule. A always pair with U and G always pair with C. Complementary you have to check. See here. C there, G there, A there. So C complementary, G sir. G sir means G, CG. G there then sir, C. A there, U. G, C, U. Search here, where G, C, U there? Here there, see. G, C, U. Means this tRNA will come here and joins. Clear? Second thing. Now this is a one tRNA there having anticodon UAC. Can you tell what will be the complementary to U there then? A sir. A there then U. C there then G. A U G where present? A U G. A U G here there. A U G means this will go to first codon. G C U it will go to array. It will go to second. Okay. Now here I am having one tRNA with the anticodon AAU. AAU. You will tell this. AAU will go to which codon? 1, 2, 3, 4. 1 and 2 already over. 3, 4, 5. You will comment. To which codon this anticodon will fit? Use this rule. Base pairing rule. Okay? Till here any confusion? What to remember? Here translation we are talking about. What is translation, sir? mRNA bringing coded message for amino acid. Ha na. mRNA bringing coded message. So this coded message which mRNA is bringing, this coded message is having number of codons. Thousands of codons are present on mRNA. So thousands of codon is present on mRNA. According to that, tRNA will bring amino acids. Amino acid. Who is bringing? All tRNA will bring. But sir, correct how there? Correct amino acid how to find out? Easy there, no? But just now we discussed that each tRNA is having anticodon. Each tRNA is having anticodon complementary to codon of mRNA. Each tRNA is having anticodon complementary to codon of mRNA. So as this matching, then your amino acid will come. This is only translation there. Read here. tRNA bringing. What it is bringing? Amino acid. For that tRNA, what is there? tRNA is having anticodon. tRNA is having anticodon. Complementary to whom? Sir, complementary to the codon of mRNA. In this way, 1111 amino acids is added. This whole process we called as a translation. You understand why it is called translation? See now, here this is one language there. AUG, GC, UU, like that, one language there. Then suddenly it will give information about amino acids. Then amino acids also one language there, no? Methionine, leucine, isoleucine, glutamic acid, valine. Suddenly language changes. AUG, this language goes, amino acid language starts. Change in language is called translation. From primary we are learning this. Language changing then it is called translation. Therefore they use this word, translation. Okay? What to remember? Sir, mRNA is having thousands of codon. As per the codon of mRNA, tRNA will bring the amino acids. As per the codon of mRNA, tRNA bringing amino acids. But how correctly added? For this purpose, tRNA is having anticodon. We are going. For this purpose, tRNA is having anticodon complementary to codon of mRNA. For this purpose, this tRNA is having anticodon complementary to codon of mRNA. This whole process we called as translation. So once your translation completes here, then 1 amino acids will come continuously. Now suppose this is your first amino acid came with this tRNA. This pink is the second amino acid comes with second tRNA. Then this amino acid also we have to join, no? Why to join, sir? At joining then only chain will form, no? Because amino acid chain is protein there. So many amino acid, first amino acid, second amino acid, third amino acid, fourth, fifth, like that, thousands are there. You have to join. So if you want to join, then something bond needed. This bond we called as a peptide bond. Remember, amino acids are joined together by peptide bond. Sir, this peptide bond who will make? For this purpose, inside this ribosome, this background blue color looking to you, this is ribosome there. For that, inside this we are having one special RNA called as rRNA. rRNA, my sir? R means ribosomal. This RNA is present in the ribosome. Therefore, it is called as rRNA. Knowing, no, in 9th standard only you completed this concept. Again asking that. rRNA is a ribosomal RNA present in the ribosome. What is role of that? It will join many amino acid. It will join by which bond? Peptide bond. Okay there. Many amino acids are bonded together, joined together by special bond called as a peptide bond. And that peptide bond who will make? It will made by sir rRNA, ribosomal RNA. Okay sir. Till here we understood. Understood no? Now tell this. First question. What is function of mRNA in protein synthesis? mRNA knowing no? Tell me. Whether mRNA stores amino acids, whether it brings the coded message from DNA, it breaks the peptide bond, or it produces the ribosome. What will be the answer? Easy then, no? So, from DNA to ribosome, mRNA is bringing message. So, in the function of mRNA, we have to write it brings the coded message from DNA to ribosome. mRNA will bring the message. Second, see the code for one amino acid on mRNA. Code for this. So the code for one amino acid on mRNA, no, AUG, together code there. This is one code there. This is second code there. What you call this code? Anticodon, gene, triplet codon or chromosome? What sir? Easy there sir. 1, 2, 3. 3 there means it is a triplet codon. Okay? Okay sir, no problem. Sure. Next, the complementary sequence present on tRNA. This is your mRNA there. On mRNA, just now we discussed on mRNA, what is it? mRNA is having codon. mRNA they are not asking, they are asking for tRNA. Complementary sequence present on tRNA. To this complementary, what is this? UAC. What is it? UCA is codon? No. Anticodon? Yes, it is an anticodon there. Peptide bond, what is it? Peptide bond is bond between two amino acids there. That only there, see now. Amino acids are joined together with the help of which bond? First amino acid, second amino acid joined by one bond. Second, third joined by another bond. What is this bond? James bond. What is the name of this bond? Check. Hydrogen bond? No sir. Glycosidic bond? No sir. Peptide bond? Yes sir. It is a peptide bond there. So amino acids are joined together with the help of peptide bond. Joined together by peptide bond, which is made by rRNA. Okay sir. Now see here. Once one amino acids will come, first amino acid came then second. These amino acids are joined together to make a protein. Right there? And this whole process where happening, it is happening in the ribosome. Please see now. Suppose here one amino acid came, one tRNA comes here, tRNA comes, then it will add one amino acid. Once the ribosome read this AUG, then ribosome will move to next. See here, AUG, then next codon what is GGG. So you see now ribosome here there in this area. It is reading AUG. So once the ribosome read first codon, same like your textbook, first page reading, then we'll go for second page, no? Same there. So AUG reading over there, ribosome will move to next codon, GGG. Then G also, GGG also reading over, then ribosome will move third codon. It will go 1, 2, 3, same only there. Only I am showing position of ribosome. Or don't think that 3 different there. Same only there. First ribosome here there. GGG. CGC. Like that. It will go on. So who is moving here? mRNA? No. mRNA same there. Same position there. Ribosome move. So ribosome move from one end of mRNA to another end. Yes or no? First ribosome was at this end. Then it started to move. Like that goes to that end. Simple there. Ribosome move from one end of mRNA to another end of mRNA by distance of one triplet codon. One triplet codon, only it will move. It is not moving that half like this, then suddenly goes here. Pattern there is that. Like this we are doing. Anyhow, on ground running hour, mRNA it is. On mRNA systematic needed, no, everything. So ribosome move from one end of mRNA to another end of mRNA. How? By how much distance it will move? It will move by distance of one triplet codon. So ribosome was here, now here, now here. Location changing, no? Location changing. Therefore it is called as, this process is called as translocation. What is word they are using? They are using the word translocation. Trans means to change, location changing. Getting? So what is translocation? Ribosome keeps moving from one end to another end of mRNA. Ribosome keeps moving from one end to another end of mRNA by distance of one triplet codon. First this codon, then move to next codon, then move to next codon, like that. So ribosome keeps moving from one end to another end of mRNA by distance of one triplet codon. One time, one triplet codon it will move. This is called as translocation. Now once all amino acids are added like this, amino acids added, one chain will form. Now this chain, one more chain will join. Again their structure will change, and finally we are getting a complex protein. How complex protein will form? Many amino acid chain together make a complex protein. Single they are simple, many together complex. Easy? So in this way you complete the complex protein synthesis. So in this whole process, what there? First transcription, information from DNA copied to RNA. Second, translation. According to mRNA, ribosome will add the amino acids with the help of tRNA. Then protein will form, many proteins will come together to make a complex protein. Clear? Children, one last concept you have to understand about the mutation. Just now we discussed about sickle cell anemia. How there it is? Suppose I am having one sequence here. Now suddenly if this sequence change means what? For example, see, here it is there, CTG there. Can you see this? CTG there. If this CTG becomes GGG, CTG sequence there, instead of that it becomes GGG. So this change in nucleotide there, no? First which nucleotides were there? CTG. Now which they became GGG. Sudden change in nucleotide like this. This change can be anyhow, means this can be deleted only. This area can be deleted. Then again it will join. Something new will be added in this. In this sequence, something new will be added. Are you getting? Change there, no? But change can be in any way. New nucleotides can be added. Old one can be deleted. Old one is replaced by another one. Anything possible. So any sudden change in the nucleotide. Any sudden change in nucleotide. For example, see here, GAT there. So originally it was DGC. So sorry, TGC. So this TGC is removed. In that place GAT came. This also changed there. So that only I am telling. New will be added. Old one will be deleted. One is replaced by another. Something change will happen in the sequence. So such sudden change in the nucleotide sequence creates problem to you. Problem sometimes very minor there. Minor means simple there. For example, if something change happen like that, you will find say some children are having extra finger one. Extra. Are you getting? Instead of 5 they are having 6. Mutation there it is. Minor change there. Some are having very considerable change also possible. Considerable means lifetime pain will be there. Just now we discussed sickle cell anemia, color blindness, that in 9th standard when it was there, no, albinism, where hair becomes white, skin becomes pale, eyes pink. Recall that all diseases possibility there because of mutation. Are you getting? So mutation what there? Sudden change in nucleotide which can cause minor or considerable change. Getting? So how to define it? Any sudden change that occurs in nucleotide sequence. Here like this. TGC instead of that GATM. This is change there. So any sudden change in the nucleotide sequence of gene. What will happen because of that? It can cause minor means small change, that 6 finger type, or considerable change. Considerable change means sickle cell anemia, color blindness, albinism, like that. Lifetime something disease you will get. So this process is called as mutation. What is mutation? Any sudden change in the nucleotide sequence of gene. Any sudden change in nucleotide sequence of gene which causes minor or considerable change in character. Minor means understood, minor means small change, something will happen in your body, or something major will happen that lifetime you will suffer because of mutation. So mutation is not good. 1, 2 example learn of mutation. 1, sickle cell anemia. We just discussed. If your RBC normal there, it is biconcave there. Biconcave. Biconcave means like this shape will be there. Okay? RBC, normal RBC. If mutation happens, then your RBC will change the shape like this sickle. Sickle understanding or no? Like this there to cut the grass. So like that you will see the shape of RBC. This is normal RBC there, it is sickle cell there. Okay? Why happen this? Genes nucleotide sequence changed. Getting? So mutation can lead to minor changes also, like this dangerous considerable change also possible. Is it clear? So if they ask you what is mutation, generally they will ask objective type of question on this. What is mutation? Any sudden change in the nucleotide sequence. Any sudden change in the nucleotide sequence which causes minor or considerable change in the character. Now 2 minutes here focus. In March 20, board question I am talking now, PYQ there it is. March 20 they asked one question to you. Transfer of information from a molecule of DNA to mRNA, what it is called? What is the answer for this? Information from DNA transferred to RNA, mRNA, what it is called? Whether it is called transcription, translation, translocation or mutation. DNA to RNA, obviously it is transcription. Clear? Second. Now this question asked in March 2024 question there in March paper. Minor changes occurs due to change in position of any nucleotide is term. Just now we discussed. Minor or considerable change because of what? Because of transcription? No sir. Translocation? No sir. Mutation? Yes sir. Mutation will be the answer for this. Okay. Now next one see. Sure. Now this question they asked in March 25. What is the question? Any nucleotide change, any nucleotide of gene suddenly changes, then minor changes occurs in that person because of what? Because of what? Because of translocation, disability, mutation or changes. You will answer this. Sure. You will comment in there. Comment box A, B, C, D. What will be the correct answer for this March 25 question there? Clear? I think up till now, whatever concept we completed, you understood it properly. If you like our videos, please subscribe the channel and share the link with your friends.